Hello everyone. So last week we started introducing the topics of variables, the things that can change in an experiment. This week we're going to talk more about all the processes of the scientific method. So if at any point in this video you are confused or want to rewind, feel free to just go back in the video, watch it over again until you understand the topic. So let's begin. So this is a nice visual of the scientific method. So with the scientific method, we always begin with asking a question. And that question could be anything like, how much water does a plant need to grow the tallest? What sort of soil helps a plant grow the best? Does listening to music help a person study more effectively? Any sort of question that you have based on what you see in your natural world, your observations. So once you have a question, then the next step of the scientific method is to do some background research. So research a little bit of what other scientists have already done or what people already know about that particular topic. And then from that research, you can make an educated guess or a hypothesis. So this is just a guess about what you think is going to happen. I think by watering a plant every day, a half a cup of water, it will grow more than if I watered it a half a cup every other day. Any educated guess based on your research. Then you form some sort of experiment that will test this hypothesis. So you could set up a series of plants, give them varying amounts of water, and see which one grew the tallest. If those results show that in fact half a cup of water every day helped that particular plant grow the tallest, then that would be your conclusion. So the hypothesis would be true, and we would be able to report the results as true. If our hypothesis was not correct, maybe a third a cup of water every other day helped the plant grow the tallest, then our hypothesis was either partially true or not quite true. So then we can go make a new hypothesis, set up a new experiment that tests that new hypothesis even more clearly, and then draw results from that experiment, and if they're correct, report those results, and if not, we can go back again. So science is always changing. If you don't get something right the first time, that's okay. It's just important to understand why your hypothesis wasn't supported, and then we can go back and make changes to our hypothesis and to our experiment so we can hopefully get the correct results the next time. Now let's look at an example. So we said before that an experiment begins usually with some sort of observation. And what observation means is the act of noticing or describing events or processes in a careful, orderly way. So looking at something, noticing something in a careful, orderly way. So if we're looking at these two patches of grass here, so we have location A and location B, just by observing, we can see that location A, the grass grew much shorter than it did at location B, right? So that would be an observation. Once we have made our initial observation, we can then make inferences based on that observation to make a prediction or our hypothesis. So if the observation for this example was that location B had much taller marsh grass than location A, our inference could be that we know something at location B is making that grass grow taller than at location A. So that would be our inference, our logical interpretation based on what we already know. We know that the grass is taller at location B, so we can infer that something in that location is making that grass taller than the other location. So from that inference, we can then begin to develop our hypothesis or our educated guess about why location B is growing taller than location A. So again, for example, researchers inferred that something limits grass growth in some places. So that would be location A. So something is limiting how tall this grass can grow in location A, whereas in location B, nothing is limiting the grass growth because the grass is growing nice and tall. So based on the knowledge of salt marshes, the scientists hypothesized that marsh grass growth is limited by available nitrogen, because scientists know that nitrogen, which is an element in the soil, helps plants grow. The plants need that nitrogen to grow taller. So that would be something that was inferred by the scientists, so they can now make their hypothesis that maybe location A does not have as much nitrogen in the soil as location B, which is why the grass is growing much taller in location B. Once a hypothesis is made, then it's important to design an experiment that tracks the variables. So the variables in this example would be the amount of sunlight the grass gets, the type of soil, the temperature, the amount of light that it gets, availability of nutrients. So the nitrogen would have been one of the nutrients in the soil that the plant is getting. All those factors that can change are called variables. Remember last week we learned about independent variables and dependent variables? So those are all things that can change. So whenever possible, a hypothesis should be tested by an experiment in which only one variable is changed. That's very important because if we change all the variables, like the light and the amount of water that it's getting, the type of soil that it's in, then we won't know which variable is causing the change, is causing the grass to grow taller. So we only want to change one variable at a time. And this is called having a controlled experiment. So all the variables that are kept the same are called controlled variables. We only want to change one. That would be our independent variable. So let's pretend that we are setting up a controlled experiment for this particular example. So the researchers would select plots of marsh grass that had similar plant density, soil type, and access to fresh water. And then we would make one our control group and one the experimental group. So as you can see the pictures at the bottom. So the researchers added nitrogen fertilizer, which we see here, N means nitrogen, and that would be the independent variable because that nitrogen fertilizer is only added to the experimental group. It's only added to one of the groups. Then they can observe the growth of the marsh grass in both the experimental and the controlled plots to see which one grew taller. Because we kept all the other variables the same, if the experimental group grew taller, we know that it's because of the added nitrogen, since that's the only thing that's different from the control group, right? Because they're getting the same amount of sunlight, the same amount of water, the same soil. Everything is the same. The only thing that's changed is the independent variable, the nitrogen, and the dependent variable would be the growth of the grass. So the thing that depends on the independent variable. So the growth, the amount of grass growth, depending on the amount of fertilizer. Then scientists can record their observations by gathering information called data. So there's two different types of data: quantitative data and qualitative data. So when we're talking about quantitative data, you want to think about quantity. So this is something that we can measure. Measurable. So that would be like the grass height, right? We can measure that by using a ruler or a yardstick. Something measurable. Whereas qualitative data, I remember it by thinking quality. Something you can describe. So if I say that the grass in the experimental group is more green than the other group, maybe I can see that the texture of the grass looks much healthier, things that you can describe using words. I apologize for my poor handwriting on this. But it's important to measure to understand the difference. So again, quantitative is usually numbers. When you think quantity, you're thinking about numbers. Qualitative, you're thinking quality. You're thinking words, something you can describe with words. Another helpful tool that scientists often use to display their data is graphs. So this is a good way, once you've already gathered your data, if we put them into a graph, we're able to visualize the difference between that data. So we can see N, again, that was nitrogen, remember? So that's the group that we added nitrogen to. And then this bottom one would be the control group. So we can see that our data for the nitrogen group, the grass grew much taller than it did for the control group. Okay, so again, a good way to visualize data is by using a graph. So to summarize about the scientific method, we start with an initial observation. So with this example, the grass was growing taller at one plot of grass in the marsh than in another part, so we wanted to know why. So then we're able to make our hypothesis, our educated guess. We think it's because some of the soil has more nitrogen than other parts of the soil. Then we designed our controlled experiment, where we added just nitrogen, all the other variables were kept the same, made observations and analyzed data. That's where we took measurements of the grass after adding the nitrogen and observing, so we did the qualitative data, quantitative data. Then we can interpret that data. So with the graph that I showed, you can see that the marsh grass with the nitrogen grew taller. Remember the graph? And then that supported our hypothesis. So we can make a conclusion that, in fact, nitrogen in the soil helps plants grow taller. So I hope this video was helpful in understanding scientific methodology, how scientists set up controlled experiments, and we'll be doing a lot of other examples in class. If you're confused, feel free to rewind this video, play it again, or email me with your questions. Okay, thank you very much.